Hello friends, today in this video we are going to discuss about the MCQs on immunology, and this is the 13th part of the series of more than 1800 MCQs on immunology with detailed explanation. Okay, and which will be beneficial for many life science exams like CSIR NET, GATE and many hospitality exams or nurse exams. Okay, and also for NEET exams. Okay, so move on to the session of MCQs on immunology. So the first question is, live vaccines are always administered parenterally or are less immunogenic or produce short-lived immunity or confer immunity similar to natural infection? And the right one is option D. That is, live vaccines confer immunity similar to natural infection. So live vaccines use a weakened or attenuated form of the germ that causes a disease. Okay, and because these vaccines are so similar to the natural infection that they help prevent, so they create a strong and long lasting immune response. Okay, so basically in live vaccines the same antigens are infected into the human body, but the antigen is weakened or attenuated, so our immune system will produce the antibodies against those antigen, but the human body cannot get symptoms of the disease because that is weakened or attenuated antigen. Okay, so move on to the next question, that is, type 1 hypersensitivity causes increased vascular permeability, causes smooth muscle construction, is immediate type of hypersensitivity, or is all of the above? And the right one is option D. That is all of the above. So what is the type 1 hypersensitivity? Type 1 hypersensitivity is also known as an immediate reaction and involves immunoglobulin E mediated release of antibodies against the soluble antigen. So this result in mast cell degranulation and release of histamine and other inflammatory mediators. Okay, so move on to the next question, that is passive immunity. So passive immunity is conferred by introduction of ready-made antibodies, is used for treatment of acute infection, is applicable in immunodeficient host, and is all of the above. And the right one is option D. It is all of the above. So what is passive immunity? So passive immunity is developed after you receive antibodies from someone or somewhere else. Okay, so that is the passive immunity. Our body didn't get do not produce their own. Okay, so that's why it is a passive, we get it from an outside. Okay, so passive immunity is also natural or artificial. Natural passive immunity is antibodies received from mother, that means through breast milk. So that is the natural passive immunity. We get it from outside but from mother, so that is a natural passive immunity. And the artificial passive immunity is antibodies received from a medicine, example from a gamma globulin injection or infusion. Okay, so this is a two type of passive immunity. So move on to the next question, that is, the following is not a foreign antigen. So enzyme, toxin, corneal tissue or dust, which one is the foreign antigen? And the right answer is option C, that is corneal tissue, that is the eye tissue, that is not a foreign antigen. Okay, so next one is secretary IgA has J chain, has a secretary component, or both of the above, none of the above. The right answer is option C, both of the above. So secretary IgA both have J chain and secretary component. So here you can see this is the J chain and this one is the this whole one, this one is the secretary component, and these two are IgA molecule. Okay, so next one is, antibody present in milk is IgG, IgM, IgE or IgD, and the right one is option A, that is IgG. So antibody present in milk is IgG. So next one is, monoclonal antibodies are the most specific antibodies formed by fusion of mouse myeloma cell with spleen cells, or both of the above, none of the above. The right answer is option C, that is both of the above. So monoclonal antibodies are most specific antibodies and formed by fusion of mouse myeloma cells with spleen cells. Okay, So here how we can produce monoclonal antibodies. So at first we have to inject the antigen into a mouse, and mouse raise the antibodies against those antigen. So we have to collect those immune cells or antigens, and then we have to fuse those immune cells with the tumor cells. Okay, and after fusion we get a hybridomas. So this is the hybridomas. So why we add this, why we fuse this immune cell with the tumor cell, because tumor cell continuously produce the antibodies or immune cells. Okay, same type of immune cells, that is the monoclonal. So this hybridoma screened for production of desired antibody. So antibody producing hybridomas cloned, and clonal expression form the monoclonal antibodies. Okay, so next one is Freund's complement adjuvant contains water in oil and dead mycobacterium, or oil in water and dead microbacterium. And the right answer is option B, that is water in oil and dead microbacterium. So adjuvants, what is adjuvant? So adjuvants are non-specific stimulator of the immune response. So when mixed with an antigen or immunogen, adjuvant help to deposit or sequester the injected material, thereby helping to increase antibody response. Okay, and Freund's adjuvant is a solution of antigen emulsified in mineral oil and used as a immunopotentiator or booster, act as a booster. Okay, so what is the features of Freund's complement adjuvant? It also called complete Freund's adjuvant, water in oil emulsion containing killed mycobacterium, greatly enhances immune response to immunogen. Okay, so it acts as a booster for antigen antibody complex formation. Okay, so next one is Freund's complement adjuvant increases cellular immunity only, humoral immunity only, both humoral and cellular immunity, or antigenicity. And the right answer is option C, both humoral and cellular immunity. Okay, so next one is immunosuppressive agents is or are anti-metabolites, corticosteroids, or uh, anti-lymphocyte serum, or all of the above. And the right one is option D, all of the above. So what is immunosuppressive agents? So immunosuppressive agents are drugs that suppress the immune system and reduce the risk of rejection of foreign bodies such as transplant organs. Okay, so move on to the next, that is exotoxins are protein in nature or present only in gram negative bacteria, or liberated only by disintegration of cell, or all of the above. So exotoxins are option A, it is protein in nature. So here exotoxins which are produced within a cell, and after that it secreted from the cell and infect the uh, humans or other animals. Okay, so this is the exotoxin. So exotoxin produced by gram positive bacteria act at specific site and produce characteristic diseases like Corynebacterium diphtheria produce diphtheria disease, and Clostridium tetani produce tetanus. So they give the exotoxins. Okay, so next one is heterophile agglutination test is or are Streptococcus mg agglutination test, Well-Felix test, both of the above, none of the above. And the right answer is option C, that is both of the above. So heterophile agglutination test is both of the above, and the test is usually performed using commercially available test kits which detect the reaction of heterophile antibodies in a person's blood sample with horse or cow red blood cell antigens, and these test kits work on the principle of latex agglutination or immunochromatography. Okay, and heterophile antigens are a type of heteroantigens that are present in two different species, but they share epitopes with each other. And what is epitope? Epitope is the part of an antigen molecule to which an antibody attached itself. Okay. So next one is neutralization test is or are Sick test or ASLO test or Nagler reaction or all of the above, and the right one is option D, all of the above. So what is neutralization test? Neutralization tests are based on the binding of antibodies to the pathogen or to toxins, resulting in the inhibition of their biological properties. And this method can be applied to detect pathogens or their toxins or to identify antibodies with defined pathogens. Okay, so next one is substrate used in ELISA is or are orthophenyl diamine dihydrochloride or paranitrophenyl phosphate or both of the above, either of the above. And the right answer is option D, either of the above. So substrate of ELISA is either of the above. So OPD or orthophenyl phenylenediamine diamine dihydrochloride is used to detect HRP and yields a water-soluble yellow-orange reaction product. The reaction product has a absorbance maxima of 492 nanometer, and PNPP, that is the paranitrophenyl phosphate disodium salt, is a widely used substrate for detecting alkaline phosphatase in ELISA application, and PNP produces a yellow water soluble reaction product that absorb light at 405 nanometers. So basically here the antigens are present, which we want to detect, and now we have to add the antibodies, and then we have to add a secondary antibody with attached with the enzyme like OPD or PNPP. Okay, and then when the substrate is added, they produce the yellow color reaction product, and we get the absorbance at 492 nanometer for OPD or for PNPP, that is 405 nanometer. Okay, so in this way we can detect in ELISA, we can detect the antigens in ELISA process. Okay, so next one is enzymes used in ELISA is or are horseradish peroxide or alkaline phosphatase or either of the above, both of the above. The right one is option C, that is either of the above. So enzymes used in ELISA is or are horseradish peroxide, alkaline phosphatase. Okay. Tumor necrosis factor R is induces fever, mediates inflammation and septic shock, or both of the above, none of the above. The right one is option C, that is both of the above. So tumor necrosis factor refers to a group of cytokines which are mainly secreted by macrophages and can induce cell death of certain tumor cell lines. Next one is TNFG is produced by macrophages or augments natural killer cell activity or both of the above, none of the above. So TNFG is none of the above. Okay. And next one is natural killer cells are derived from large granular lymphocytes or spontaneously cytolytic for certain tumor lines or involved in allograft rejection or all of the above. And the right one is option D, all of the above. So natural killer cells are lymphocyte in the same family as T and B cells coming from a common progenitor. Okay, so from a common progenitor, T cell, B cell and natural killer cell comes from them. And however, as cells of the innate immune system, NK cells are classified as group 1 innate lymphocyte and respond quickly to a wide variety of pathological challenges, and natural killer cells are best known for killing virally infected cells and detecting and controlling early sign of cancers. Okay, so next one is Arthros reaction is due to relative antigen excess or produces mainly IgE or complement activation is essential or all of the above, and the right one is option C, that is complement activation is essential for Arthurs reaction. So what is Arthurs reaction? Arthurs reaction is a localized type 3 hypersensitivity reaction. Local vasculitis develop as a result of immune complex deposition. Inhaled antigens may induce similar reaction in the lung, IgG type antibody or farmer's lung and pigeon breeder's lung. Okay, and for by this Arthur's reaction, this can be happened. Okay, this is a type 3 hypersensitivity reaction. Next one is serum sickness is generalized type 3 hypersensitivity reaction due to relative antigen excess post streptococcal glomerulonephritis or all of the above, and the right one is option D, that is all of the above. So serum sickness, what is serum sickness? So serum sickness in humans is a reaction to proteins in antiserum derived from a non-human animal source occurring 5 to 10 days after exposure, and symptoms often include a rash, joint pain, fever. So serum sickness after injecting of a foreign serum or certain drugs, antigen is excreted slowly leading to antibody production. So antigen and antibody complex produced, this complex may circulate or be deposited at various sites. Symptoms: fever, urticaria or lymphadenopathy, and symptoms develop after few days to 2 weeks. And serum sickness is classified as immediate reaction due to the fact that symptoms develop promptly after immune complex are formed. So it produces like this. Serum sickness can be seen like this. So the next one is tuberculin test is a subcutaneous test or read after 24 to 48 hours or uses purified protein derivative of Mycobacterium bovis, or cells involved are lymphocytes, monocytes or macrophages, and the right one is option D, that is tuberculin test where cells involved are lymphocyte, monocyte and macrophages. So tuberculin skin test is a skin test to determine past or present infection with the tuberculosis bacterium based on hypersensitivity of the skin to tuberculin. And here, after infecting the tuberculin into our hands, then it produce a sensitivity test like this. Okay, so next one is contact dermatitis is a type 4 hypersensitivity reaction, is detected by patch testing on skin or involves infiltration of macrophages or is all of the above. And the right one is option D. This is all of the above. So contact dermatitis is an itchy rash caused by direct contact with a substance or an allergic reaction to it, and the rash isn't contagious, but it can be very uncomfortable. So many substances can cause this reaction, such as cosmetics, fragrances, jewelry and plants, and the rash often shown up within days of exposure. So this is the contact dermatitis seen like this. Okay, so next one is HLA typing is done by mixed leukocyte culture test or lymphocyte transfer test or cytotoxic test or all of the above, and the right one is option D, that is HLA typing is done by all of the above. So HLA, that is a human leukocyte antigen typing, is a kind of genetic test, and HLA typing is sometimes called HLA matching. So HLA typing looks at certain factors related to the immune system. The test can help figure out if a person can safely donate bone marrow, cord blood or an organ to someone who needs a transplant. Okay, so next one is BCG is made up of bacterial products or live viral vaccine or killed viral vaccine or live bacterial vaccine, and the right one is option D. So BCG is a live bacterial vaccine. So Bacillus calmette-guerin vaccine is a vaccine primarily used against tuberculosis. It is named after its inventors Albert Calmette and Camille Guerin. In countries where tuberculosis or leprosy is common, one dose is recommended in healthy babies as soon after birth as possible. Okay. So last question is DPT is made up of bacterial product, or live viral vaccine, or killed viral vaccine, or live bacterial vaccine? And the right one is option A, that is made up of bacterial products. So DPT vaccine or DTP vaccine is a class of combined vaccines against three infectious disease in humans, that is diphtheria, pertussis, that is the whooping cough, and tetanus. And the vaccine components include diphtheria and tetanus toxoid, and either killed whole cells of the bacterium that causes pertussis or pertussis antigens. Okay, so these are all about this uh, part 13 MCQs. Okay, so thank you for watching this video. So we will see you on our next video, that is part 14 of immunological MCQs. Okay, thank you for watching this video.